Hello everyone, I'm Barry Jones from Oxford Instruments Magnetic Resonance, based in Oxford in the UK. I would like to welcome you to our webinar on NMR for core analysis. In this webinar, I will first give a brief introduction to the technology of NMR measurements on rock cores, and then give some practical examples about how it is used. First, let's look at what measurements we can make with NMR. The fundamental measurement we make is about pore geometry, in particular the pore size distribution. From this, we can derive parameters such as porosity, irreducible fluids, and free fluid index. We can also calculate permeability, get estimates of wettability, and calculate capillary pressure. Finally, NMR measurements and core plugs can be used to calibrate downhole NMR well logs. Let's start by looking at how we generate NMR signals from rock core samples, and then how we use them. When we excite a liquid sample with a radio frequency pulse or series of pulses, the NMR signal we see coming back from the sample is generally an exponential decay, governed by a characteristic time constant called T2. T2 depends on the nature of the liquid and to some extent on its environment. Free fluids, for example, generally have longer T2s than bound fluids. For fluids in confined spaces, such as in the pores of a rock core plug, the relationship between T2 and the pore size is governed by this equation. The T2 of the bulk fluid, T2 bulk, is relatively large, and TE is an instrumental parameter that we can make small. So the first and the third terms of this equation disappear. What we are left with is a relationship that shows that T2 is inversely proportional to S over V, or in other words, the pore size. So, the T2 from fluids in large pores is relatively long and gets shorter and shorter as the pores go from large to medium to small. It's also important to note that NMR measurements come only from the fluids in the pore spaces, such as water or oil. There are no signals from empty spaces or from the rock matrix itself. In this sense, NMR measurements are complementary to CT or X-ray. CT and X-ray give information about the solid structure of the core and tell you nothing about the fluids in the spaces. NMR, on the other hand, gives you information about the nature of the fluids and their environment in the core, but tells us nothing at all about the structure. Of course, in a real rock sample, we can't pick and choose which pores to examine. We can only see a total signal from all of the pores in the sample. Some large pores with long T2s, some medium-sized pores with medium T2s, and some small pores with very short T2s. The resulting signal is a composite of all the signals from the pores in the rock. In theory, one for each different pore size. What we need is some way to go back from this composite signal to work out how much of each separate T2 signal is in the composite signal. This will tell us something about the spread or the distribution of T2s in the sample. Fortunately, there is a way to do this. It's a mathematical procedure called inverse Laplace transformation, or ILT as it's commonly known, or sometimes just inversion. This process breaks down the composite signal into all of the different T2 signals that went into it, with a weighting for how much of each is there. Typically, the composite signal is broken down into about 256 separate T2 values, or bins, which are then shown as a T2 spectrum or distribution. Because we know that short T2s come from small pores and long T2s come from large pores, this T2 distribution is in effect a pore size distribution. In the example shown here, we can see that the sample is composed mainly of long T2s or large pores. Here is an illustration of how large and small pores appear in the raw NMR signal and after inversion. Large pores, as in this blue trace, have a relatively long T2, which after inversion shows up as a high peak at the top end of the T2 or pore size scale. Small pores, from the red trace, on the other hand, have short NMR T2 signals, and the inversion shows no peaks at this high end of the scale. Now let's look at how the T2 distribution measurement is used in practice to give us hard information on our cores. Here is an example of a typical T2 distribution that you might get from a laboratory NMR core measurement. You start by first cleaning and then saturating your core sample, usually with brine of the same salinity as the reservoir. Then you place the sample in the NMR instrument and run a scan. After inversion of the data, you will see a display similar to the dark red line shown on this chart. The light red line is simply the cumulative value of this saturated T2 distribution. You then remove the sample from the instrument, centrifuge it to irreducible water saturation, and rescan it. This time you get a trace like the blue one shown here, with the light blue line being simply the accumulation of this desaturated distribution. From these traces, we can now derive several important parameters of the core sample. The final value of the light red line, the cumulative value of the saturated distribution, is the total pore volume, or porosity. The final value of the light blue line, that is the area under the desaturated distribution, gives us the bound volume. The difference between the two is the free fluid. The T2 cutoff, which marks an approximate separation between bound and free fluid, is the T2 value where the accumulated saturated distribution intersects with the final cumulative bound volume value. The next slide will show how the T2 cutoff is used to calibrate NMR well logs. The usual practice when evaluating NMR well log data is to use the same T2 cutoff values for the whole dataset to calculate bound and free fluid volumes. However, this may not be the best way to do the calculation, as the T2 cutoff value may well vary at different depths in the well. A more accurate way to do the calculation is to take core plugs from multiple depths in the well, then perform the laboratory T2 measurements on the plugs and retrospectively apply the appropriate T2 cutoff value for each depth. This will lead to a more accurate assessment of free and bound volumes. This slide shows a typical set of core analysis results as presented in the instrument software. The box in the yellow ring here shows the summary results that we've talked about in the previous few slides. The red trace shows the raw data from the saturated plug, and the blue trace shows the data from the plug after centrifuging. Using the basic T2 distribution, we can go on from here to model permeability, using either the logarithmic mean of the distribution, known as the Schlumberger method, or the ratio of free to bound fluid, also known as the Coates method. In both cases, the user is free to apply their own preferred value of the various constants, such as A and B, in here. Now let's look at some slightly more advanced applications without going into too much detail. We will take a brief look at wettability, capillary pressure, and two-dimensional data maps. First, let's look at wettability, and let's start by looking at a water-wet rock during an oil flood experiment. For this measurement, we need to first saturate the plug with D2O instead of ordinary water, so that we can separate the signals from oil and water. The deuterium nucleus in D2O does not give an NMR signal, so we know that the signals we see must be coming from the protons in the oil. At the start of this measurement, water, or in this case D2O, coats the pore walls, with oil in the center. This means that the oil T2s will be relatively long, as the oil is not interacting with the pore wall surface. In the T2 distribution, we will see a small peak at a relatively long T2. As we push more oil into the rock during the flood, the oil peak increases because there is now more oil, and starts to spread out towards lower T2s as the oil approaches and begins to interact with the pore walls. In conclusion, for a water-wet rock, the oil distribution peak gets larger and spreads left towards shorter T2s. Now let's look at an oil-wet rock, where the opposite happens. At the start, the oil T2 distribution will be concentrated at short T2s, as the oil is in contact with the pore walls. As we push more oil into the rock, the oil peak increases in size and starts to spread out towards longer T2s as more oil moves away from the pore walls. In conclusion, for an oil-wet rock, the oil distribution peak increases and spreads right towards longer T2s. In summary again, for an oil-wet rock, the oil T2 distribution increases and spreads right. For a water-wet rock, it increases and spreads left. This slide shows a real-life example of the oil flood measurement we just looked at. The different color traces shown on this graph are oil T2 distributions from the same sample as we successively push in more oil. The distribution goes from black to green to blue and finally to red. As you can see, the oil peak increases and moves to the right, indicating that this rock is oil wet, not water wet. Now let's look at how NMR can be used to measure capillary pressure. The capillary pressure at any point R in a sample being spun in a centrifuge is given by the Hassler-Brunner equation, where delta rho is the density difference between the wetting and non-wetting fluids, omega is the centrifuge rotation speed, and R0 is the distance from the centrifuge axis to the outlet face of the core plug. The traditional method for measuring capillary pressure by centrifuge first calculates the capillary pressure at the inlet face by substituting Ri, the distance to the inlet face, into the Hassler-Brunner equation. We then calculate the average saturation of the core plug at each spin speed and use that calculation to estimate the saturation at the inlet face. We calculate the average saturation by collecting the amount of fluid expelled from the plug at each spin speed. Measuring the expelled fluid requires a graduated collector and a stroboscope synchronized to the rotation speed of the centrifuge so that the graduation scale can be read. This process has several sources of error, both in the measurement of the expelled fluid and the estimation of the inlet saturation from the total average saturation. This process is repeated, typically at 8 different spin speeds, each one lasting for a day, and thereby producing 8 data points over a period of about 8 days. By contrast, the NMR method makes no estimations about the saturation at the inlet face of the core plug. The NMR method divides the plug into 30 to 40 virtual slices and measures the actual saturation in each slice by a simple MRI technique called a 1D profile. There is no need to collect the expelled fluid, so no need for a graduated collector or stroboscope. Consequently, a relatively simple centrifuge can be used. For each virtual slice along the length of the core, we calculate the capillary pressure using the Hassler-Brunner equation, and we measure the saturation of the slice using the MRI 1D profile method. We therefore have a set of points to plot against each other on the capillary pressure curve. For each spin speed, we have 30 to 40 slices for which we can calculate capillary pressure and measure the saturation. So we need only 2 or 3 spin speeds to cover a wide range of capillary pressures and collect 60 to 90 data points for the PC curve. This graph shows a set of data points obtained by the NMR centrifuge method using 4 spin speeds over a period of 4 days. The yellow points here are from the lowest spin speed. The blue points are from the highest spin speed. You can see that when the data points for all 4 sets are plotted together, they agree very closely with the results from a traditional porous plate method shown here by the purple squares. So to summarize, the traditional method for capillary pressure measurement by centrifuge measures the expelled fluid and then estimates the inlet saturation for each of about 8 spin speeds. This gives us 8 points collected over a period of about 8 days. The NMR method, on the other hand, measures the saturations at up to 30 positions for each of 3 spin speeds, giving us 90 data points over a period of only 3 days. The NMR method allows the fluids in the core and surrounding it in the holder to be reversed in order to generate both drainage and imbibition curves. The final subject of this webinar is 2D data maps, which are being used more and more to help solve difficult problems, such as where more than one fluid is present in the sample at the same time. As the name suggests, these methods take 2 sets of data from the same sample, using different pulse sequences, and combine them together. The algorithms for doing this combination and doing the 2D inversions are provided either by our partners GIT or by license through us from Schlumberger. The first example uses measurements of the 2 NMR relaxation properties T1 and T2 to help with fluid typing. Gases generally have long T1s and short T2s, whereas water is the opposite. Plotting these 2 parameters against each other therefore helps to separate the gas and the water. Another 2D method is to plot T2 against diffusion. Different fluids may have similar T2s but different diffusion constants. Finally, this method uses a variation of the T2 measurement to give information on the connectivity of pores in a sample. The final example of an NMR application for core analysis in our webinar today is a fairly advanced one using spatially resolved T2 distributions. In a similar way to what we talked about earlier with capillary pressure measurements, we can divide a core sample virtually into slices. But in this case, instead of measuring saturation in each slice, we collect a T2 distribution in each slice. This slide shows a time sequence where brine is flooded from the bottom into a decane saturated core. We can see that the predominant T2s move to the left or shorter values as the flood progresses, indicating that the fluid is moving into the smaller pore spaces. So that is the end of our webinar for today. I thank you all very much for watching and hope you have learned something that you didn't already know about NMR rock core measurements. Now I'll be happy to try to answer any questions you may have.